Hello and welcome. In this video, we are going to discuss the syllabus and the topics of genetics. Genetics is certainly a very important part of paper 2. If you notice the last 15 years question, you will observe that genetics along with breeding, biotech and biostats accounts for the largest share of questions in botany paper 2. The syllabus is short if you look at genetics in isolation, but along with breeding, biotech and biostat, the syllabus becomes considerably big and it takes some time to complete all the topics. The syllabus is largely clearly laid out except for a few hidden topics which we will identify. The purpose of this video is to make you identify the subtopics within the individual topics, identify some of the hidden topics, and finally identify the prospective question areas. Like all other videos of this series, in the end I'm also going to tell you about what will be a good source to study genetics in terms of textbooks. I'm going to suggest you an Indian book and also an overseas author book. Depending on your comfort, you can choose one of these books to study your genetics. No matter how you plan to take the paper 2, genetics will be getting maximum amount of attention and maximum amount of time in preparation. But the time and energy spent in genetics will be paying back very, very handsomely in your examination. Now let us start the discussion and let us see how the UPSC has given the syllabus of genetics. In the next few slides, I will be restructuring the syllabus for a conceptual coherence when it comes to coverage. And then we will start with individual topic analysis. This is how the syllabus looks like. If you want, you can pause the video at this point of time and you can take some time to read the syllabus. However, this syllabus looks a little jumbled to me because there are a number of topics which should be placed close together, are not placed close together, and then some disjointed topics have been placed together. Apart from that, there is another aspect that there are a few topics which have been just repeated in different words. So this is my reconstructed version of genetics. First and foremost, we need to be clear that genetics, the syllabus can be divided into 3 parts as the UPSC gives it. The first part will be transmission genetics. Transmission genetics is also known as classical genetics. This is where we study about how the genetic transmission happens, how does it manifest at the level of phenotype, and how these ratios can be modified by different genetic phenomena. In total, there are 15 topics in transmission genetics. If you look at it, first topic is development of genetics, which is nearly a dead topic because hardly any question has been asked, and it is a very, very vague topic that's been mentioned. At the most, you can study a little amount of historical development of genetics as a discipline of biological science. But after the topic 1, every other topic is pretty clearly laid out, and the questions have been both direct and also from inside the topic. There is one hidden topic, that is Mendelian inheritance. Hidden because this is not mentioned in the syllabus. I have added it from my side, but question on Mendelian inheritance have been asked in the past. And another thing that is important to note is that there are a number of topics which cannot be understood well if we have not done Mendelian inheritance. Look at topic number 5 and topic number 6, incomplete dominance and multiple alleles. Both these topics need a certain basic understanding of Mendelian genetics. If we do not understand that, it will be very difficult. Similarly, when we come to linkage and linkage analysis, they are the most important factor behind deviation from Mendelian ratios of independent assortment. Again, to understand the importance of linkage and how does it modify the phenotypic expression in offsprings, it is important to have a base of Mendelian genetics. Hence, I have added Mendelian genetics from my side. Other than that, every other topic is mentioned in the syllabus. I might have merely reordered the syllabus. So let's give a quick reading to the syllabus and then we go to the topics of molecular genetics. So development of genetics is one topic, gene versus allele concept, then pseudoalleles, Mendelian inheritance, incomplete dominance, multiple alleles, quantitative genetics, polygenic inheritance, and multiple factors. They are mentioned in a little fragmented manner in the original syllabus given by the UPSC, but they all need to be bundled together. Then you have to do linkage, crossing over. After linkage and crossing over, gene mapping, and this will also include molecular maps. Topic 11, 12, and 13 are related to sex determination. It starts with sex chromosome, then the mechanisms of sex determination, and the syllabus also makes a mention of molecular mechanism of sex differentiation. Finally, the syllabus also talks about sex-linked inheritance. The last two topics, 14 and 15, are about cytoplasmic inheritance. So you have to study cytoplasmic inheritance, cytoplasmic genes, basically the organellar genes, and 15 topic is a manifestation of cytoplasmic inheritance, that is genetics of male sterility. This is one of the foundational topic to understand plant breeding, which you will be exploring as a next section in your botany syllabus. After this, there are a few topics, 6 to be precise, in molecular genetics. Topics are less, but they are actually a little big topics. If you look at the first topic, it is mutations, biochemical and molecular basis. Mutations is a big topic because there are many subtopics which come under it, and questions have been asked from these subtopics. After this, structure and synthesis of nucleic acids and proteins. Genetic code is a small topic, but topic 2, this one, the structure and synthesis of nucleic acid, is a big topic. After that, regulation of gene expression is also a big topic. The next two topics are relatively small. Gene silencing is essentially a manifestation of regulation of gene expression, primarily in eukaryotes. And finally, multigene families. This is also a short note, and questions which have been asked on multigene family have been pretty straight. After that, there are 3 topics related to organic evolution. One is evidences of organic evolution. Next is mechanism and theories of organic evolution. And the third topic is rather small, that is role of RNA in origin and evolution. So only 3 topics, but here topic 2 is slightly big because there are a few subtopics involved here. Before getting into individual topics and identifying their subtopics, let's first and foremost understand what kind of weightage has been given in the examination to genetics. This chart makes it clear. This chart I had used in my previous video also, which I had made on cell biology. And by the way, I'm expecting that you have already completed cell biology, because from your emails I get to know that a number of students are following this series very diligently, and some of you are working very hard to keep up with the calendar which I am suggesting, which is extremely commendable. Now, coming to the weightage, if you notice, in paper 2, the largest chunk of questions are coming from genetics, breeding, biotechnology, and biostats. If you treat it as one group, it accounts for 40% of questions in the last 15 years. So that's quite a significant weightage. The second largest area is plant physiology and biochemistry, which we will be dealing in our subsequent videos. My next video will be on plant breeding, biotechnology, and biostats, and then further next video will be on plant physiology and biochemistry. We have already done cell biology, and the last area will be ecology and plant geography in paper 2. So ecology and plant geography gives you only 12% of total questions asked in paper 2. That is what the last 15 years analysis. But this weightage can shift a little from year to year, but this is the long time scale average weightage that's been given. One thing is very clear, that genetics is a very important area and it cannot be ignored. Now let us get into the individual topics of genetics. As I was telling you, the first topic, that is development of genetics, is kind of a dead topic because hardly any question has been asked. So I would say that a chronology of landmark developments in the field of genetics should be known to you. Other than that, this topic has no further scope, and this is exactly why there has been no question asked on this topic in the last 10 years, or for that matter in the last 15 years. You can safely ignore this topic and you can come to the real scientific topics of genetics, and it starts with gene versus allele concepts. First and foremost, here you have to do the modern concept of genes. You can keep writing with me. So the first topic is the modern concept of genes. And here you will have to give how the term gene was coined and how, despite not using the term gene, Mendel used the term factors, and how the term factors correspond to the concept of genes. Then how the biochemical concept of gene evolved from one gene, one enzyme hypothesis to one gene, one polypeptide hypothesis, how central dogma modified it, and finally the genomic studies and our modern day molecular understanding of gene expression. How does it define the gene properly? So if you look at the modern day definition of gene, so gene will be a well-defined or sometimes overlapping or a nested segment of DNA or RNA that encodes at least one fundamental effector molecule in a cell. Now, why I'm using the word RNA? Because there are a number of viruses where the genomic material is RNA. So gene can be a well-defined segment of RNA. Why I use the term overlapping? Reason is obvious. Within the bacteria, there are a number of genes which overlap with each other. What is nested gene? I leave it to you to find it out, but nested gene phenomenon is present in a number of eukaryotes. So gene can be nested, it can be well defined, it can be overlapping, but it is always going to give rise to one fundamental effector molecule. That means it can be a polypeptide, or it can be an effector RNA like ribosomal RNA, transfer RNA, enzymatic RNA, silencing RNA, small cytoplasmic RNA, pvRNA, whatever. So if a segment of DNA or RNA is capable of encoding one of the fundamental effector molecule, it will be called a gene. But mind it that genomic studies show us that there are a number of genes which can give rise to multiple fundamental effectors. That means one gene can give rise to more than one protein, one gene can give rise to more than one RNA, and that all needs to come under the modern concept of genes. And then you have to explain how do the genes function. That means the expanded version of central dogma. And once you have mentioned both of these, your gene concept answer is proper. Now, alleles are different forms of the same gene. Here you will need to explain how different alleles of a given gene occupy the same locus on a given chromosome, how alleles arise, how in organisms alleles behave to control the phenotype. All of these you need to mention. A short note can be expected from gene concept, and a short note is possible on alleles. So this needs to be prepared as one short note, and gene concept is another short note. So modern concept of genes, then alleles, where you discuss what are alleles, then how do they arise. After that, their mode of functioning and control of phenotype. Two short notes and this topic is completely done. Next topic is also a very small topic, pseudoalleles. So what happens in case of pseudoalleles, that these are having the allele type of nucleotide sequence, that means very similar sequence compared to the original gene, and that ways they may function like an allele, but there is a fundamental difference that pseudoalleles do not occupy the same locus as the alleles occupy. In this topic, what should you be prepared for? You should be prepared for what do you understand by pseudoalleles and how do they differ from alleles. This is the question, the answer of which should be answered by you, the answer of which should be satisfactorily given by you, and apart from that, I have not seen any significant question deviating from this theme. When you do this, you are done with this topic. The subsequent topics are going to be a little bigger. Mendelian inheritance. In Mendelian inheritance, there are three things that you need to be prepared for. First, experiment of Mendel, in which you should be covering a little of history. Then you need to cover choice of experimental organism, design of experiment. How did Mendel design his experiment? What were the features Mendel was studying? Then findings of monohybrid crosses and dihybrid crosses. Second topic that you should be preparing is Mendel's laws of inheritance. That means the law of segregation, the law of dominance, and the law of independent assortment. Third that you need to know is deviations from laws of Mendel. With these three topics, you would have satisfactorily done Mendelian inheritance. Apart from that, there is nothing else which is required. Let's remind ourselves that Mendelian inheritance is not a clearly stated topic, but to understand further topics, it is important. At the same time, sometimes direct questions have also been asked. The next topic is actually a deviation from Mendelian inheritance. The format that you adopt for incomplete dominance, same format of preparation you can adopt for the next topic, that is multiple allelism. First and foremost, understand that you are only going to be asked a direct question where first you have to explain the phenomenon, then examples, then its genetic and molecular basis, its impact on Mendelian ratios. Lastly, significance. And it is exactly the same set of topics which you have to prepare for the next topic. The next topic, as you notice, is multiple allelism. And in multiple allelism also, you have to mention the phenomenon, the examples, genetic and molecular basis, impact on Mendelian ratios, and significance, and you will be completely doing both these topics in complete dominance. And here it is, multiple alleles. Okay, so what I'm noticing is that both the titles are same, incomplete dominance and again incomplete dominance. But what I mean here is not incomplete dominance, but here I mean multiple alleles. Both the topics need to be covered under similar subtopics. Next is quantitative genetics and multiple factors. And another term which I have not mentioned here for the constraint of a space is polygenic inheritance. So what happens that there are certain traits which are governed by not one gene but a number of genes. And now number of genes will present a bigger number of alleles. And the final manifestation of the trait is going to be the result of the interaction among these many different alleles. Here you would not have a binary choice. For example, in Mendelian inheritance, the plant was either tall or dwarf. The flower color was either purple or it was white. So it is not going to be binary here. Here you are going to see a continuous gradation in the feature. Think of skin color, think of the body mass of humans. So you will notice that there is no binary relation here. You will find all sort of body mass in human beings, all sort of skin tone in human beings. Why? Because these traits, and of course many traits in plants, cattle, are governed by several genes and their multiple alleles. And this is exactly why it is called quantitative inheritance rather than qualitative inheritance. What are the topics that you should be preparing here? First and foremost, the phenomenon. What is it? How does it manifest, etc. Then difference from Mendelian inheritance, its operation, examples, and importance. So this is going to be your preparation for a general topic, a general question, general or broad question. This is the most common question that has been asked, and once in a rare while there will be a short note on this aspect. So let's write comparison with Mendelian inheritance. Prepare it for 200 words. Prepare it for approximately 300 words. Most likely in the examination, you will get an opportunity to write about 200 or 250 words, but it is always safer to prepare it for approximately 300 words. Next topic is linkage, an important topic. There are 3 questions which can be asked to you in linkage. First, explain the phenomenon of linkage. How does it affect the genetic transmission ratios, etc. So here the format will be explain the phenomenon, then its basis, its types, its detection and its significance. The second question on linkage can be on its impact on inheritance ratios. The question can be variously worded, but the scope will be that how, if linkage is operating, it will cause a deviation from the expected Mendelian inheritance ratios that you would normally expect from independent assortment. Certainly that is not seen. So this is the second question. And third question is its relation with crossing over. So in nutshell, crossing over breaks linkage. Linkage is the occurrence of multiple genes present on the same chromosome, and because of that, these multiple genes fail to display independent assortment in the Mendelian ratios. If at all these genes get separated from each other and behave independently, this is because of crossing over. So in short, crossing over breaks linkage. That's the whole thing. But when a relation with crossing over is asked, you will first have to explain how does linkage operate, and then you have to explain how does crossing over operate and how crossing over undoes the linkage effect. The next topic is crossing over itself. In crossing over, you have to study the cytological basis, the molecular basis, and importance of crossing over. Just these three things you have to do. I'm just writing it for your easy recall. You have to first explain what is crossing over, then do a little bit of its history, discovery, etc. Then cytological basis. And in cytological basis, if you want to get an edge over your competitors, do mention mitotic and meiotic crossing over, and then in detail explain meiotic crossing over. After that, give the molecular basis. In molecular basis, in brief, describe the model of Robin Holiday, and then you should be mentioning the double strand break repair model. This is one question here. This is another question. And this introductory part can be attached to any answer. So whenever you are writing an answer on crossing over, so of course you will explain the phenomenon, and in brief you will tell about its discovery, and then you will be coming to either the cytological basis or the molecular basis as demanded by the question. If you are asked the mechanism of crossing over, in brief you will have to give both cytological as well as molecular basis. Here I am not mentioning the relation between crossing over and linkage, because in the very previous topic, linkage, I have already mentioned relation with crossing over, so there is no need to repeat the same question. So well, that is crossing over, and we are now coming to gene mapping. And the topic clearly says that you also have to study how do we prepare molecular maps of genes. So here, first and foremost, you will go for Genetic mapping. Genetic mapping is another name for linkage mapping. In this, direct questions have been asked, so prepare very well the principles of linkage mapping, 2-point test cross and 3-point test cross. And both these test crosses you have to illustrate properly. And then you will come to molecular maps. First, why we go for molecular maps? What are the shortcomings of genetic mapping because of which we have to go for molecular map? After that, what are the main methods of molecular maps? So name the method, then what is its principle, how do we operate it, and finally, in what situations do we apply it? So there are 5-6 methods of gene mapping. You have to mention all the methods of gene mapping, molecular gene mapping, and in each one, very briefly, you have to mention the principle by which it operates, then how do we operate it as such, and finally, under what situations do we apply this. After this, we come to sex determination related topics. So first start with the mechanisms of sex determination, and you have to understand that majority of the studies on sex determination have been done on animal species. But even in plants, significant research has been done, and in all the categories where you find the mechanism of sex determination with a plant example, it will be important to mention the plant example. First and foremost, what we have to study in sex determination. Define sex determination nicely. After that, there are 2 major mechanisms of sex determination: genetic sex determination and environmental sex determination. Explain the different genetic mechanisms of sex determination. It will be single gene based sex determination, sex chromosome based sex determination, and autosome based sex determination. There will be subtypes of sex chromosome based sex determination. All the types need to be explained by you, like XX, XO, then XX, XY, then ZZ, ZO, ZZ, ZW, and then genic balance in Drosophila. After this, environmental sex determination, how the environmental context or the temperature at a specific time of development will determine the sex of the organism. These are ESD or environmental sex determination mechanism. You have to deal with both of them. After this, you will come to sex differentiation, the molecular mechanism. In plants also molecular mechanism are known, and in Drosophila, sex differentiation molecular mechanism has been excellently studied. That also you have to mention. So I'm quickly writing what I just now explained to you. So in sex determination, first definition, then mechanisms. In mechanisms, genetic sex determination and environmental sex determination. In genetic sex determination, monogenic sex determination, sex chromosome based sex determination, and autosome based sex determination. Then sex chromosome based, you will be dealing with XX, XO type, XX, XY type, ZZ, ZO type, ZZ, ZW type, genic balance and compound sex chromosome phenomenon. Autosomal sex determination you will find manifesting as ploidy variation based sex determination, and then in ESD the context dependent sex determination and temperature dependent sex determination. In sex differentiation you have to deal with plants, and excellent example is found in Drosophila. So the entire mechanism should be done. Next topic is sex-linked inheritance. This is a fairly straight and simple topic. What is sex-linked inheritance? What are the types? Then its operation, and finally its significance. These things you should do, plus differentiate it with sex limited traits and sex influenced traits. With proper examples, you will find these examples from the animal world, but you have to do it. After this, you come to cytoplasmic inheritance. Cytoplasmic inheritance takes place because of organellar genome, and to be precise, mitochondrial and chloroplast genome. In cytoplasmic inheritance, what is cytoplasmic inheritance? What is the basis of cytoplasmic inheritance? What are the salient features of cytoplasmic inheritance? How do we detect a case of cytoplasmic inheritance? How does it differ from maternal inheritance? Not maternal inheritance, I'm sorry, maternal influence. Then examples from the plant world. This is the first thing which will be catering to a general question, which is the most common question, but on mitochondrial genome questions have been asked, plus origin of mitochondria. This I had also touched in my cell biology discussion, so you should be looking into endosymbiotic origin of mitochondria. Similarly for chloroplast also, chloroplast genome and origin of chloroplast. The last topic will be a manifestation of cytoplasmic inheritance only, that is genetics of male sterility. Although male sterility is not totally in the hand of cytoplasmic inheritance, there are other inheritance patterns also which affect male sterility. Let's take a look. In male sterility, first and foremost, have a clear definition and initial explanation. What does it include? After that, you will have to deal with genic male sterility. Second is cytoplasmic male sterility. Third is cytoplasmic genic male sterility. In each one, you have to discuss operation. How does it operate? Example. After this, male sterility using transgenics. Will also be expected from you. And finally, chemicals to induce male sterility. So these are the topics that you ought to be doing on male sterility. With this, you are completely done with transmission genetics. Next few topics are on molecular genetics. Mutations is a big topic. Here, what is mutation? Types of mutation, mutagenesis, and within mutagenesis, spontaneous and induced, and within induced, chemically induced and induced by radiation, and finally importance of mutations. So what you notice here, to be very clear what I am meaning here, these are the questions. Types of mutations. Second question is a general question on mutagenesis. Third question is spontaneous mutations. Fourth question is induced mutation, a general question. Fifth question is chemical mutagenesis. Sixth question is radiation based mutagenesis. And importance is a tail that can be added to any of the questions. Explain the types of mutation and add a note on its importance. Explain spontaneous mutagenesis, add a note on importance of mutations. So that way. Next topic is appearing very simple, but it's a big topic. First is structure and synthesis of nucleic acid and proteins. First, talk about the structure. So you have to study the structure of proteins, DNA, and RNA. Then, in case of RNA, also its types. After this, synthesis. With respect to synthesis, you have to do synthesis of DNA. That means DNA replication, and DNA replication you have to do for prokaryote and eukaryote. Then synthesis of RNA, that simply means transcription. Transcription you have to do for prokaryotes and eukaryotes. Third is synthesis of proteins. That means translation. You have to do it for prokaryotes. You have to do it for eukaryotes. To be clear, how many different types of questions we are looking at? Protein structure, one question. DNA structure, second question. RNA structure and types, third question. DNA replication, general question, fourth. DNA replication in prokaryotes, fifth. DNA replication in eukaryotes, sixth. Transcription, general question, seventh. Transcription in prokaryote, 8. Transcription in eukaryote, 9. 10th question is a general question on translation. 11th, translation in prokaryotes. And 12th, translation in eukaryotes. So you can see that within this one line, structure and synthesis of nucleic acid and proteins, how many different questions are hidden. Each question needs to be prepared by you in approximately 250 to 300 words, not more. Those who come from biotech background, you will have a tendency to study big chapters from foreign author book. But remember, ultimately you have to distill everything in 250 words. So save your time and be very, very focused on the needs of the question. Next one is genetic code. It's a short topic. So what is genetic code? Its discovery, the Hargobind Khurana experiment, then its features, then standard versus non-standard codes. That's all you need to do on genetic code. There is going to be a direct question on genetic code. What is genetic code? Explain its properties, and maybe sometime they will ask you add a note on non-standard codes. Next is regulation of gene expressions. This is again going to be separately in prokaryotes. There are 5 to 7 mechanisms which you have to do, and then you have to do in eukaryotes. Again, there are about 8 to 10 mechanisms in eukaryotes. In prokaryotes, there are certain mechanisms which can be asked as a separate question. So general question can always be asked, gene expression regulation in prokaryotes, but at the same time, you may be asked a question on operon model, and attenuation. These are the questions that might be asked to you. In eukaryotes, you can be asked on RNA interference. Of course, there is going to be a general question on eukaryotic gene expression regulation, but a question on RNA interference, a question on mRNA editing, a question on transcription level gene regulation in eukaryotes, and translation level gene regulation in eukaryotes may be asked to you. So well, this is what gene expression regulation is. There are 8 questions in this topic, as you can see it. And then next is gene silencing. Gene silencing is long-term expressional silencing of genes by mechanisms other than mutation. So obviously it is done by mechanisms of gene expression regulation and spatially at post-transcriptional level. When you are doing this topic thoroughly, that is regulation of gene expression in eukaryotes, there are certain topics which will give you an idea on gene expression silencing. There are certain transcription level gene expression regulation mechanisms which will silence the gene on the long term, like methylation, histone phosphorylation, histone, other types of histone modification. Then RNA interference, RNAi is an important mechanism of gene silencing. Then there are certain silencing complexes which bind to the genetic material and they render the gene expressionally silent. All of these mechanisms which you have learnt in this topic will have to be used in gene silencing. If you are doing eukaryotic gene expression regulation nicely, you should not be needing to study anything separately on gene silencing. After gene silencing, one topic is multi-gene families. Here you have to deal with what are multi-gene families, what are the types of multi-gene families, what is the occurrence of multi-gene families, how do they arise, so their origin. Then also discuss different types of gene homology relations. Ultimately, significance. 2 types of questions can be asked. First question is a general question where you have to deal with all the types. Second question is this: origin of gene families and comment upon different types of gene homology relations. So I guess once you are doing this topic thoroughly, you will be done. Now you are done with molecular genetics. The next three topics are related to evolution. First is a very obvious topic, evidences of organic evolution. There are so many lines of evidences in organic evolution, right from the fossil records to anatomical evidences to homology studies to molecular evidences, biochemical evidences, evidences from migration, so on. So create a nice comprehensive list with many headings, but try to summarize it with clear example within 300 words, that is evidences of organic evolution. Next is mechanism and theories of evolution. Here you should be doing Darwin's theory of natural selection, after this modern synthesis, where you deal with neo-Darwinism, Hardy-Weinberg equilibrium. Apart from that, as an added safety measure, I would also ask you to study speciation. So first do anagenic and cladogenic speciation, and then within cladogenic speciation deal with the sympatric, allopatric, parapatric, peripatric and instant speciation. Prepare this, you never know when a short note on speciation can be asked to you. Last topic here is role of RNA in origin and evolution of life. So RNA, it is at present accepted by molecular evolutionary biologists that RNA predominated the earth in pre-cellular days. This is the molecule which was in abundance. It carried information and at the same time it could have carried out catalytic activities. This notion became strong and finally it took the shape of RNA world hypothesis given by Walter Gilbert. So when we look at RNA, it is agreed that it was perhaps the earliest complex macromolecule capable of self-replication, carrying information both. So you will have to deal with RNA world hypothesis, the evidences for RNA world hypothesis, the properties of RNA because of which it could have been the very first molecule, and finally, what are the shortcomings of the RNA world hypothesis. This is an important topic. And here you have to do RNA world hypothesis. What is it? What are the properties of RNA which give credence to RNA world hypothesis? Then the sequence of events according to the RNA world hypothesis, and then certain shortcomings. So well, this was the core syllabus of genetics, molecular biology and evolution. We are done with that. The next question is, what should you be studying for it? I have got two books. One book I had recommended last week in the context of cell bio. So if you already have that book, then this book will help you for genetics also. That is Cell Bio, Genetics, Small Bio, Evolution and Ecology by Dr. P.S. Verma and Dr. V.K. Agrawal. It is brought out by S. Chand Publication. So this book will help you with most of the topics of genetics, small bio and evolution. And if you are more comfortable studying international authors' book, then Concepts of Genetics by Klug and Cummings. Klug and Cummings will be an excellent source to study. Any one will be a good source, or if you want to have everything in a concise exam-oriented form, then you can consider my study material, which is available as a part of my guided self-study program or my online course. So if you need concise at one place all the topics, then my study material will be a good solution. So any one of these three sources, my study material, Klug and Cummings or P.S. Verma, wherever you feel most comfortable, I would say adopt that source and do this extremely interesting and highly rewarding area of paper 2 of Botany Optional. So on that note, I will leave you for this week, and next week I will be discussing the syllabus of breeding biotech and certain molecular methods and ultimately biostats. So till then, goodbye and good luck to all of you.